The title of the talk today is Human Social Genomics, Recent Findings and Clinical Implications, with an emphasis on the recent findings and forecasting toward implications. And this is just a real honor for me. It's the first time that I've been able to attend this meeting. I'm already looking forward to coming back next year. Applause just for that. That's pretty good. Let me say, my primary focus is how does stress affect health? Not just what are the effects, but how do experiences of stress get under the skin to influence biological systems that are relevant for health? Why do I care about this so passionately? Well, you may have seen this Institute of Medicine report just recently showing that life expectancy for American adults is last for males and second last to females compared to other individuals in 16 other economically comparable countries. This is really atrocious. The same findings emerge if you look at disease-related mortality. We're not first or second or third in terms of economically well-developed countries. We're second to last, and the only country that's worse than us is Denmark. We are number one in infant mortality, number one in adolescent obesity, and number one in heart disease. We're winning, guys. We're winning. We're doing it. Not only do we have high rates of disease burden mortality, but there are two things that are particularly disturbing to me. See if this disturbs you as well. First, these data, this is for the Commonwealth Fund, a great report if you haven't found it. Commonwealth Fund 2014 on their website. We have the most expensive healthcare system in the world, but also one of the least effective. You probably already have some indication of this. Our average healthcare expenditures per capita is the highest, but we are last or nearly last with respect to healthcare access, efficiency, and equity. You can see our rankings on the right there. Number 11, number 11, number 11, number 11, number 11. $8,500 per head for annual healthcare costs. We pay more and we get less. What's the second disturbing thing to me? Out of every 10 deaths in the United States today, in the world today, 7 are caused by conditions that can be prevented or minimized. Depression, Alzheimer's disease, certain types of breast cancer, obesity, diabetes, and heart disease. Now, based on this picture, you're probably thinking, got it, I'm totally with you, George. Let's reduce physical inactivity. Let's get physical activity up. Let's reduce alcohol use. Let's reduce smoking. It's a very interesting meta-analytic review of a bunch of different studies published in PLOS Medicine in 2010. They just looked at the odds ratio of all-cause chronic disease-related mortality as a function of these traditional risk factors and, in addition, high levels of social stress. Social stress compared to these traditional risk factors was a stronger predictor of chronic disease-related mortality than physical inactivity, excessive alcohol use, smoking, and a bunch of other things that I'm not showing here. Now, social environmental patterning of mortality risk is not totally new. That's not a new concept. 20 years ago, House and colleagues published a really interesting and landmark paper in Science showing that across multiple countries and cultures, mortality risk decreases as social integration increases. And this is cross-cultural, this is internationally. As best we can tell, social integration is really important for health. Social isolation and other types of social stress are really bad for health, especially for health that involves immune system activity, and I'll talk about that in a second. We have found similar effects in some of the research that we've done. This is a study of 168 women with histologically confirmed epithelial ovarian cancer. We observed them over a period of 7 years. The best predictor of longevity in this particular study, when everything shook out, social connection. After 7 years, 63% of women who are socially well connected were still alive compared to only 36% of women who are relatively socially isolated. Social stress alone, in this analysis, accounted for 27% of the variance that differed in women who lived longer with epithelial ovarian cancer versus women who died more quickly. Considered together, these findings, they point to two really interesting and important questions for us to consider as doctors, as researchers. Number one, how does social stress reach so deep inside of the body? I mean, in this case, we're talking about cancer biology, right? So it's presumably not just that social stress or social integration is just leading to negative thoughts or negative emotions. I mean, it's probably doing that. But if we see these effects in certain models, we start to wonder, well, could it be the case that social stress is actually influencing something like the tumor microenvironment or the immune system? How deep is it that social stress actually goes? How deep can we see these signals? And number two, we know that social stress is implicated not just in the experience of depressive episodes and anxiety episodes, PTSD, experiences of social stress and rejection, one of the best proximal predictors of heart attacks, epithelial ovarian cancer. I just gave you an example. So how can it be that social stress exerts such broad effects on health? Why would that be the case? Okay, so answers to these questions, we think, can be viewed through the lens of an exciting new field called human social genomics. This is a paper. This is work that's in collaboration with Steve Cole, one of my good colleagues at UCLA. Here's the general idea for human social genomics. We're all walking around the world, and we notice our moods change. We notice our thoughts change, right? Sometimes we're in a good mood and a bad mood. We can monitor those things. We have direct access to our thoughts and our moods. We don't have direct access to what's going on with our immune cells or what's going on inside our immune cells. So more so, we learned growing up that our DNA is endowed from our mother and our father, and the prevailing societal view is that we're relatively biologically stable, right? We can't access changes. We can't feel changes necessarily in our immune system, certainly not in our human genome. So because of societal influences and because we don't have access to those changes, we've come to the very reasonable assumption that we're relatively stable biological entities, socially, colloquially speaking. What's interesting about human social genomics, which the human part started in 2007, the animal stuff goes back a little bit further, is that recent studies have suggested that literally hundreds of genes in the human genome can change their transcriptional stance, can change their activity as a function of the different physical environments that we find ourselves in, and also the different social environments that we find ourselves in. And what's most interesting is out of the 40,000 or so transcripts that we can look at in order to look at the activity of the human genome, we don't see changes in just any random sets of genes as a function of experiences of social stress. What we've seen so far, and again, the entire field is only about 15 to 20 studies, but what we have seen so far is that social stress is particularly strongly linked to changes in the expression of genes that are involved in innate immunity, antiviral response, and et cetera. And I know you want to hear more about that, and that's what the rest of the talk is about. So we're very interested in understanding and in further documenting exactly what types of stress, what types of social stress drive this process, and how do these effects occur mechanistically. This is one study we conducted. This is with a sample of 147 adolescent girls who are at elevated risk for depression, mean age is 17. They don't have any current or lifetime history of depression, but they have elevated dysfunctional attitudes or negative beliefs about themselves and about other people. We followed them for 2.5 years. We brought them into the laboratory every 6 months. Every time we brought them into the laboratory, we diagnosed them to see what depressive symptoms they had, whether or not they met criteria for clinical depression or not. And at every 6 months, we also took a blood sample so we could use that information, do the genome-wide transcriptional profiling on them, and also use real-time PCR and some other methods to see what types of genes were upregulated versus downregulated as a function of experiencing different types of recent life events. So these are all within subjects analyses. The analyses are asking what types of genes are differentially expressed as a function of what you just experienced compared to what you're going to experience during the next session when you're brought in. What we found was we were very interested from prior data which showed that a particular class of stressful life event called targeted rejection seemed to be the strongest proximal risk factor for depression. Targeted rejection events, very simply, are experiences where somebody breaks up with you, or where you're fired at work, where there's a very clear intention to break the social bond. the risk factor for depression following those experiences is about 21.6-fold increase over baseline. Compared to other types of major life events, like losing a job but where it's company-wide layoffs, the risk factor is actually only about 2-fold increase in risk for depression. So we see almost a 20-fold increase over and above other types of life stress for these particular targeted rejection life events. So back to the point here, though. What we found was that at visits where these participants, these adolescents, experienced a recent severe life event that involved targeted rejection, they exhibited higher quantities of mRNA for NF-kappa B and inhibitor of kappa B compared to visits when no such stress occurred. Now, these effects we see for both NF-kappa B on the left and inhibitor of kappa B on the right. We find them for both transcription factors, and we only find that these effects are specific to targeted rejection forms of life stress. So if you just look at any type of life stress in general, you don't see this structuring of transcription factor activity as a function of stress. It seems to be particular to interpersonal life events and to forms of social stress that involve the severing of a relational tie or some information about how intact the external social world is and how friendly that social world is. We did a replication of this study, slightly different participant sample. This is 121 children, aged 9 to 18, diagnosed with asthma in the past, no acute respiratory illness, no chronic illness other than asthma. In this study, we followed them for 2 years only, as if this is really easy to do. It's tough work to do, to have no attrition over this amount of time. Followed them for 2 years, brought them in every 6 months. We interviewed them to find out their asthma symptoms, also all of the stressful life events they experienced over the past 6 months. We also took the blood samples again at each of these time points. We looked particularly at levels of mRNA for the glucocorticoid receptor alpha and glucocorticoid receptor beta, because we think that these transcription factors regulate localized inflammation in the airway pathway, can lead to airway constriction and the exacerbation of asthma symptoms. What we found was a very interesting interaction between the experience of recent targeted rejection life events and the extent to which individuals viewed themselves as being high versus low in social status. So the first message is that individuals had lower levels of glucocorticoid receptor alpha, which downregulates inflammation in the airways, if they experienced a recent targeted rejection life event compared to if they didn't. So we see that same effect, in this case, experiencing more recent social stresses associated with less anti-inflammatory transcription factors that would then implicate less inflammatory gene expression. But who does better under those circumstances of social rejection? Is it the case that girls who are, in this case, all women, girls who are lower in social status, do they do worse when they experience social rejection? Or is it the people at the top of the social hierarchy? Well, we assess this using a ladder. It's literally a ladder on a page, and you ask people to just identify in their social network where do they stand on the ladder compared to other individuals in the social network. If you do that in this analysis, what we found was that actually individuals who rated themselves as being highest in social status had the greatest reductions in anti-inflammatory transcription factors when they experienced targeted rejection. So it could have been, so what we're saying here is that those, at least in adolescence, the high status people had the furthest to fall. In-term summary. Social adversity, we know, very strong independent risk factor for a lot of different diseases, all-cause mortality, specifically mortality related to inflammatory conditions. Social adversity, we think, may exert these effects by skewing the activity of the leukocyte basal transcriptome toward a more pro-inflammatory, quote, threat preparedness state, and away from an anti-inflammatory state. And we see evidence of these effects for broad categories of social stress, such as socioeconomic status and social isolation, and for specific types of stressors, such as targeted rejection life events. Now, you might say, okay, that's fine, George. Most of those studies are all correlational. I mean, you know, there's lots of things that could be contributing to the gene expression findings. Do we have any experimental evidence that social stress really influences the activity of the immune system or the genome? So to examine whether these regions might also be implicated in experiencing social pain and perhaps in regulating inflammatory response, we brought a set of about 30 of these participants who did that public speaking task back into the laboratory, and we had them do something in the scanner. They showed up with 2 other individuals. We laid them down in the scanner. We told them that the other individuals were going to 2 other adjacent fMRI scanners. We gave them a button box that they put on their right hand. These were all right-handed because it's fMRI and we need to be consistent with that. And we told them every time you receive a ball, you can throw the ball to either of the other 2 participants, Jennifer or April. And the participant is at the bottom. So everything is great, right? You're playing this ball tossing game, life is wonderful, you know, your parents love you, everything is good. But what happens is that when you're laying dark in the scanner with nobody around you, but you think you're playing this ball tossing game with 2 other people, halfway through the game, all of a sudden, you stop getting the ball, leading to an experience of social isolation and rejection. Can you imagine yourself in a scanner thinking that you're playing this ball tossing game with 2 other people? Okay, what do we find? We find that this brief experience, this is only a 2-minute experience of social isolation and rejection, reliably engages parts of the brain that are involved in experiencing physical pain. The dorsal anterior cingulate cortex and the anterior insula, which I'm not showing here. But believe me, it also lights up. And if you look at the extent to which individuals show this neural response in these brain regions, and use that neural data to predict the inflammatory responding to the public speaking task, what you see is that the more neurally sensitive you are to social evaluation and rejection, the stronger inflammatory response you have to the public speaking task. Okay, so we didn't focus on gene expression there, but we focused on cytokines. So presumably, we would see those same effects represented at the level of gene expression as well. We're looking at that now. Considered together, these findings have begun to elucidate what I've called a social signal transduction pathway that regulates gene expression and ultimately influences health. From this perspective, experiences of social stress get represented in the brain in a network of brain regions that some people have called the physical or social pain network or the threat network. They involve the dorsal anterior cingulate cortex, the anterior insula. Now, these higher level brain regions don't have the ability to directly regulate peripheral physiology, but they do project to lower level brain regions that do regulate the sympathetic nervous system, the HPA axis, such as the PAG, the autonomic control nuclei, et cetera, et cetera. Those brain regions directly regulate the SNS and the HPA. The end products of those axes, we've already talked about, epinephrine, norepinephrine, cortisol. Those factors interact with receptors on immune cells. Those signals can then get transduced to transcription factors. Those transcription factors can translocate into the nucleus of the cell, where they can bind to certain transcription binding sites, influence transcription of DNA into RNA and translation of RNA into proteins such as pro-inflammatory cytokines, which then go throughout the body and do their job, which is really great in the short term and really bad in the long term. You want your immune system to go to 6th gear when you have a viral or a bacterial infection. You don't want it to go to 6th gear all the time. Now, let's just take another perspective on what we're talking about here, right? The other perspective is, wait, why the heck would the body be doing this at all? Right? Why do we see, I mean, it sounds counterintuitive, right? Why would the body do anything that would essentially kill the body off, right? Well, we don't know. But here's some speculation from where we stand, where I stand. Historically speaking, up in the left-hand corner there, The innate immune system developed to counter physical threats from predatory animals and dangerous conspecifics. Those are the things that dominated our ancestral environment. An inflammatory response to these physical threats is critical, right? Why? It accelerates wound healing, limits infection, but an even more adaptive response, immune system response, is an anticipatory response in advance of an actual physical insult. One of my favorite examples of this is a study of knee surgery that was recently done at Stanford. They brought some men in for knee surgery as normal, other men for knee surgery, but they stressed them out before they went in for knee surgery. Who had better clinical outcomes? The men who were stressed out before they came in. Why? Because you see this redistribution of immune cells all throughout the body. So once the body is wounded, the immune cells are already there to do their thing. A preparatory or an anticipatory immune system response may have evolved and may have involved monitoring the social environment for potential cues indicating higher risk for physical threats, such as being alone and not being able to protect yourself, which may have enacted what we've called increases in pro-inflammatory gene expression, reductions in antiviral immunity. Why? Well, because if you're socially isolated, you're not likely to get a virus from somebody else. Increases in inflammation, decreases in antiviral immunity. We've called this the conserved transcriptional response to adversity, CTRA. Now, what's the issue here? There are two issues. First, we don't often experience physical threats in the contemporary social environment. I mean, we hear about them all of the time on the news, but it's not often that we personally are directly facing a tiger or out in the wilderness and we're alone, right? So that's one problem. The second problem is that this entire model that I described on the previous slide, all of these stress axes and pathways, can be activated and engaged just as strongly by purely imagined experiences of social stress in the absence of actual physical danger. So we can activate this entire axis leading to inflammatory gene expression in response to social stressors that have not yet occurred and that may never actually occur. We need to measure the genomes of many more people to understand exactly what are the most important and influential aspects of the social environment that have the ability to regulate the human genome. We've focused on some of them, but we've only started with the cast of characters that we know are huge risk factors. But also we need to measure the human environome. We need to have data, much more data, on people's life stress histories so we can understand over the life course what types of experiences are most negative for health and for human gene expression. I just have 2 more slides, so let me just mention that so far we've only talked about the individual. And you've probably been thinking, well, maybe this is just an individualistic issue, but have you ever sat next to somebody who's really anxious? Yeah? What happens? Yeah, you kind of get anxious yourself, right? So what's the implication there? The implication is that if you're anxious, you may not just be regulating the activity of your own genome, you may be regulating the activity of the genome of the person next to you. We are physically disconnected, but we are transcriptionally connected. And if that's not reason enough to care about human gene expression, consider this. About 2% of your entire molecular makeup must be renewed every day to replace dead cells and decaying proteins. And as a result, the experiences that you have today in this room, in the next room, later tonight, whether you're angry or sad or happy, will influence your molecular composition for the next 2 to 3 months. So in closing, be good to your molecular self, to the molecular selves of the people around you, and to each other. Thank you. This is the website. All the papers I've discussed are on here.